Good day. This is Psychology 311, Study Session 14. Chemical senses. 2. Gustation. Taste. The receptors for taste, called taste buds, are situated chiefly in the tongue, but they are also located in the roof of the mouth and near the pharynx. They are able to detect 4 basic tastes: salty, sweet, bitter, and sour. The tongue also can detect a sensation called umami from taste receptors sensitive to amino acids. Generally, the taste buds close to the tip of the tongue are sensitive to sweet taste, whereas those in the back of the tongue are sensitive to bitter taste. The taste buds on top and on the side of the tongue are sensitive to salty and sour tastes. At the base of each taste bud, there is a nerve that sends the sensation to the brain. The sense of taste functions in the coordination with the sense of smell. The number of taste buds varies substantially from individual to individual, but greater numbers increase sensitivity. Women in general have a greater number of taste buds than men. As in the case of color blindness, some people are insensitive to some tastes. Mechanism of taste and the gustatory receptors. The receptors involved in gustation are found in specialized end organs called taste buds, embedded in the epithelium that covers the surface of the tongue, soft palate, pharynx, larynx, and epiglottis. However, they are not uniformly distributed in these regions. The taste buds on the tongue are associated with characteristic papillae, from the Latin for pimples, whereas those in the other regions are found on the smooth epithelial surface. In humans, the number of taste buds varies considerably from person to person, with the majority having 2,000 to 5,000 distributed over the various regions. However, the number can be as low as 500 and as high as 20,000 in some individuals. The papillae in different regions of the tongue have distinctive shapes and characteristic numbers of taste buds associated with them. Scattered over the main body of the tongue are approximately 200 small, mushroom-shaped, fungiform papillae, which have, on average, only 3 taste buds each. Larger foliate papillae are found at the back and sides of the tongue. They consist of up to 9 folds of epithelium and have many as 600 taste buds each. 8 to 12 larger mushroom-shaped circumvallate papillae, each surrounded by circular throat, lie at the back of the tongue in a V-shaped formation. These have on average 250 taste buds each. Scattered taste buds are also found in the epithelium of the soft palate, pharynx, larynx, and epiglottis. Each taste bud is contacted at its base by the terminal of sensory nerve fibers. These taste fibers belong to 3 different cranial nerves connected to the brain. The nerve supply for most of the taste buds on the soft palate and towards the front of the tongue come from a division of the 11th facial cranial nerve called the cauda tympani. Because it routes to the brainstem passes close to the tympanic membrane in the hair. The ninth, glossopharyngeal and tenth vagus nerves innervate taste buds in the back of the mouth and the pharynx respectively. Each taste bud contains 50 to 150 neuroepithelial receptor cells arranged like segments of an orange to form a compact, pear-shaped structure. There is a small 2 to 10 mm opening in the epithelial surface called the taste pore, which allows direct contact between chemicals dissolved in the saliva and the tips of the receptor cells. These exposed parts of the receptor cells are made up of many long corrugated folds in the membrane called microvilli, which provides a greater surface area for contact with the saliva. It is difficult to taste food with a dry mouth. Saliva is essential for normal taste because it acts as both a solvent for the chemicals as well as a transport medium for those chemicals to reach the receptors. A layer of saliva extends into the taste pores and constantly bathes the receptors. The dissolved chemicals diffuse through this thin layer of saliva to reach the microvilli. Chewing, taste, and smell to varying degrees stimulate reflex secretion of saliva from the salivary glands under the tongue and in the cheeks. As demonstrated in Ivan Pavlov's classical experiment on dogs, the simple form of unconscious learning known as conditioning. The reflex secretion of saliva to the familiar signs of an impending meal, the sound of a dinner bell, the clatter of crockery, and the sight of the food. The taste bud complex is a dynamic system in which the receptor cells are rapidly turning over. The lifespan of an individual receptor cell is about 10 days. Cells are continually being born through the division of epithelial stem cells within the buds, maturing, performing their gustatory function, and eventually dying. Even though the receptor cell does not itself have an axon or fiber, the base of the cell has specialized regions that look like the terminals of nerve fibers. The cytoplasm in these regions is packed with tiny spherical vessels filled with a chemical transmitter substance which is released when the potential inside the receptor cells becomes more positive. Depolarization. In close association with these regions are the endings of the sensory nerve fibers, making an assembly like a synapse. Each taste bud is innervated by more than one nerve fiber, and each single nerve fiber can connect to a number of receptor cells, taste buds, and even papillae. This suggests a high degree of convergence of input from taste buds on to the sensory nerve fibers. Because of the rapid turnover of receptor cells, the connections between cells and nerve fibers are constantly changing. The nerves are continuously sprouting new processes, forming new synapses with young cells and retracting synaptic connections with dying cells. At any one time, less than a third of the cells in the taste bud are innervated. An intact nerve supply is necessary for the normal function of taste buds. If the nerves are damaged, the taste buds degenerate and slough off. And following regeneration of the nerves, the taste buds reappear. Since the time of Aristotle, 384 to 322 BC, there have been attempts to categorize taste into primary or basic taste. Although many hundreds of different chemicals can stimulate activity in taste receptor cells, the four basic taste qualities of salt, sour, sweet, and bitter have stood the test of time. However, there is still controversy as to whether combination of these four primary tastes adequately describes all gustatory experiences. Metallic and astringent tastes have, in the past, been suggested as primaries, and more recently, Japanese researchers have proposed that the characteristic taste of monosodium glutamate, used as a taste enhancer by the food industry, is also a basic taste with its own receptive mechanism. They have called it umami, meaning delicious taste. Because of the dual role of gustatory receptor cells, detecting both the nutrients and toxins, they must be able to respond either individually or collectively to a wide variety of chemicals. These chemicals range from simple ions such as sodium, salt, and hydrogen, sour, to the more complex compounds that give the sensations of sweet, for example, sugar, and bitter, for example, quinine. The mechanisms by which the chemical stimuli are translated into electrical events in the receptor cell, transduction, are numerous, varied, and complex. The essential process depends on specific interactions because taste substances and specialized protein receptor molecules embedded in the membrane of the receptor cell, which trigger a series of chemical reactions, leading to a change in the flow of ions through pores in the membranes and hence a change in the electrical potential inside the cell. However, there does not appear to be a unique mechanism for each of the basic tastes. Each seems to use several different mechanisms. There may even be similarities of mechanism for different basic tastes. The way in which we can perceive many subtle tastes and distinguish between different compounds of the same basic taste category might be explained by the multiplicity of specificity of these mechanisms. The evidence for a particular receptor mechanism is best for sweet sensation. First, certain drugs have specific effects on the detection of sweetness. For instance, after eating a West African fruit called miracle fruit, even quite acidic substances such as lemon juice, which would normally be sour, taste extraordinarily sweet. Miracle fruit contains a substance that is thought to attach to the protein receptor molecules that detect sweet-tasting substances. A subsequent increase in acidity in the saliva is thought to alter the binding of this substance with the sweet receptor protein such that it stimulates the receptor like a genuine sweet substance. In contrast, gymnemic acid found in an Indian plant, Gymnema sylvestre, blocks the sweet receptor in some manner and abolishes the sensation of sweetness for half an hour or so. Very recently, a gene called TIR2 has been identified in mice, which are selectively switched on in taste bud receptor cells. It turns out that strains of mice that lack sweet taste, they don't prefer sweet food to non-sweet, have a mutation of this gene. There is a very similar gene in human beings. Researchers have recorded with tiny electrodes from individual nerve fibers innervating the taste buds in anesthetized animals. One might have expected that each fiber will respond with a burst of impulses when a solution of just one of the primary taste substances was dripped onto the appropriate taste buds or buds. Such selectivity of response is, in fact, very rare. Most nerve fibers respond to two or more of the basic taste stimuli, the magnitude of the response varying from one taste substance to another. In other words, the activity of such a fiber does not provide unambiguous information to the brain about the nature of the stimulus. At some point, the brain must perform a comparison between the activities in several different nerve fibers in order to decide what the taste actually is. The signals from the taste buds are relayed via a chain of nerve cells and fibers at various cell stations in the brainstem and thalamus up to the cerebral cortex. Some experiments in monkeys suggest that nerve cells at higher levels in the taste pathway respond more selectively, with a larger proportion of them essentially responding to only one basic taste. At the first relay in the brainstem, almost no neurons respond to one taste, yet in the taste area of the cortex, about 75% of neurons respond to a single taste. The common chemical sense is the sensation caused by the stimulation of free nerve endings by potentially harmful chemicals. The evidence suggests that the free nerve endings are polymodal nociceptors, receptive nerve endings that respond to mechanical, thermal, and noxious stimulation. Amongst the chemicals that are known to stimulate these receptors, besides noxious, damaging chemicals are alcohol, menthol, peppermint, and capsaicin chili pepper. Summary. In this lecture, we have discussed the tongue and noted that taste papillae can be seen on the tongue as little red dots or raised bumps, particularly at the front of the tongue. We noted the mechanism involved in taste and the functioning of gustatory receptors. We also examined the 5 basic tastes: salt, sour, sweet, bitter, and umami, and the various taste areas of the brain contributing to the behavior in different ways. The hypothalamus apparently releases taste to hunger and satiety. The final destination of a taste stimulus is the taste area in the parietal lobe of the cerebral cortex, where taste is perceived. This is the end of study session 14. Thanks for listening.